Welcome to zoology class. In the last class, we have been discussing about Mendelian disorders due to single gene mutation. And in the last class, we have discussed about hemophilia. What is hemophilia? And the pattern of inheritance. So all those things we have discussed in the last class. Now we will discuss, in this class, we shall discuss another example for Mendelian disorder, that is sickle cell anemia. So the term is sickle cell anemia. The disorder is sickle cell anemia. Now let us see what is anemia. So you must have heard about this term, anemia. Anemia is a condition or a set of conditions produced in the body as a result of low hemoglobin. So low amount of hemoglobin in the blood can be of different reasons. That may be low production of hemoglobin, low synthesis of hemoglobin in the body, or lower count of RBCs in the body because hemoglobin is present in the RBC. Hemoglobin is a red colored pigment that is present within the RBC in human being and in mammals. And that the low amount of hemoglobin can also be due to less number of RBCs in the blood. And another reason for low amount of hemoglobin is less volume of blood. When blood is lost as a result of an injury or something like that, the blood volume may be reduced. And in that case also, hemoglobin may be low in the blood, and that may lead to anemia. Okay. Hemoglobin, the function of hemoglobin is to transport oxygen and to some extent carbon dioxide. So transport of these respiratory gases is the major function of hemoglobin. So when the amount of hemoglobin becomes less in the blood, oxygen carrying capacity of the blood would be reduced. That means cells, tissues and organs would receive low amount or inadequate amount of oxygen. So that would affect very seriously the vital organs of our body like brain, heart and other muscles, tissues like muscles, etc. And the all-round performance of the body will be reduced. So that may lead to dizziness, tiredness, muscle fatigue, and a lot of other problems may be resulted due to the inadequate amount of hemoglobin in the body. So this is anemia. Now what is sickle cell anemia? That's the point of our discussion here. What is sickle cell anemia? You know what is sickle? Okay, it's a weapon used by farmers. Sickle. Sickle cell. Here cell means RBC, red blood cell. So RBC becomes, the shape of the RBC becomes sickle shaped. It becomes sickle-like. It appears sickle-like. So normally the RBC should be in mammals. The RBCs are spherical, biconcave and enucleated. You know that. It should be spherical. A healthy normal RBC should be spherical in shape. But in the case of sickle cell anemia people, the RBCs become sickle shaped. And that is due to the presence of an abnormal hemoglobin in RBC. So RBC carries hemoglobin. When it carries a normal hemoglobin, it is spherically shaped. So there is a relationship between the structure of hemoglobin and the shape of the RBC. Normal hemoglobin is situated within a spherical RBC, and an abnormal hemoglobin makes the shape of the RBC sickle shaped. So we should know what is an abnormal hemoglobin. So before going into the abnormality of the hemoglobin, we should know what is hemoglobin, what is the structure of the hemoglobin in brief. Hemoglobin is a pigment. It has two components mainly, a heme part and a globin part. The heme is an iron containing chemical group that's called porphyrin. The details you did not study. So each porphyrin group has an iron. Such 4 porphyrin groups are there. And this is the heme part. So 4 iron atoms are there in a hemoglobin molecule that carries the oxygen. And this heme is connected with a globin protein. And this globin protein is in the quaternary stage of the protein. So in the first year you had already studied different stages of protein. Primary stage, secondary stage, secondary level, primary level, secondary level, tertiary level and quaternary level. So quaternary level is applicable for proteins which has more than one polypeptide chain. So here the globin of hemoglobin has 4 polypeptide chains. And each of these polypeptide chain is in the tertiary stage. And when these 4 polypeptide chains in the tertiary stage are present in a protein, that level is called quaternary stage. Quaternary stage of protein. So globin in hemoglobin is in the quaternary stage, where each of the 4 polypeptide chains is in the tertiary stage. And out of these 4 polypeptide chains of the globin, 2 are known as alpha chains and 2 are known as beta chains. So there are 2 alpha chains and 2 beta chains. They are polypeptide chains. Okay. So this is the normal hemoglobin, and the polypeptide chain is made up of amino acid. For each and every polypeptide chain, there is an amino acid, there is a specific amino acid sequence. So here also, for these alpha chains and beta chains of the globin protein, there is a specific amino acid sequence. They are made up of amino acid. The beta chain of the globin protein in hemoglobin is made up of 146 amino acids. 146 amino acids. And there is a specific amino acid sequence for this beta chain. And in a normal beta chain of hemoglobin, of globin protein, the first 7 amino acid sequences valine, histidine, leucine, threonine, proline, glutamic acid and again glutamic acid. So this is the amino acid sequence up to the 7th amino acid in the beta chain of a globin protein in hemoglobin. Let us say once again, the first amino acid is valine, the second one is histidine, the third one is leucine, fourth one is threonine, and the fifth one is proline, sixth one is glutamic acid, and seventh one is also glutamic acid. And the amino acid sequence goes on like this up to the 46th amino acid, and the rest of the sequence is not here for our discussion here, so I am not mentioning it now. Okay, so this is the sequence of amino acid up to the 7th amino acid for a normal beta chain, for a normal beta polypeptide chain in globin protein. But in sickle cell anemia person, the beta chain is abnormal. It is not normal. Hence the beta chain is abnormal. The entire hemoglobin becomes abnormal and it becomes unable to carry oxygen. Now let us see what is the abnormality. Where lies the abnormality? So abnormality comes in the 6th position or 6th amino acid. You have seen the 6th amino acid is glutamic acid. And in the abnormal hemoglobin, the 6th glutamic acid, 6th amino acid, glutamic acid, would be replaced by another amino acid, that is valine. So in the normal amino acid sequence, there is no valine in the 6th position. Instead of that, the original one is glutamic acid. But in the abnormal hemoglobin, the beta chain carries or possesses valine in the 6th position instead of glutamic acid. How does it happen? It happens as a result of mutation. Mutation of a gene located on the 11th chromosome. So it is an autosomal mutation, not sex chromosomal mutation as we have seen in the case of hemophilia. It's an autosomal mutation. So the gene for the normal gene for a normal beta chain of the globin protein is located on the 11th chromosome. And when this gene located on the 11th chromosome for the correct sequence of amino acid of beta chain gets mutated, that mutation reflects in the change of amino acid in the 6th position of the amino acid sequence of the beta chain. So as a result of that mutation of the gene on the 11th chromosome, glutamic acid will be replaced by valine. It may seem a very simple change. Out of the 146 amino acids present in the beta chain, only one amino acid is changed. It may seem very simple, but that becomes a very serious problem. That results into a very serious problem, and the hemoglobin becomes abnormal due to that change, and RBC becomes sickle shaped, and hemoglobin fails to carry oxygen, fails to transport oxygen. Okay, so this is sickle cell anemia. So when a person carries such a mutated gene, he becomes a sickle cell anemia patient. Okay, and he may exhibit all the symptoms of anemia like tiredness, fatigue, low brain function, heart problems, general tiredness, like so many other symptoms. And apart from these symptoms, a sickle cell anemia patient may have more chance of blood clotting, internal blood clotting, thrombosis, that may lead to thrombosis. So the person may be more susceptible to thrombosis also. That is another problem. So this is sickle cell anemia. Now let us discuss how this disease is inherited from one generation to another generation. For this, we have to consider a marriage between two individuals, between a man and a woman. Suppose the man is sickle cell anemia carrier. Now what is a sickle cell anemia carrier? And before that, let me say, the gene, the normal gene is denoted here as HbA, and the mutated gene is denoted as HbS. A is capital letter, S is also capital letter. So HbA, two alleles should be there. A pair of factors, as Mendel told. HbA denotes normal person, and he produces or she produces normal beta chain, normal hemoglobin. HbS is a heterozygous condition. HbS. What is HbS? HbS is the mutated allele. HbS condition is a heterozygous condition, and that person is termed as a carrier, sickle cell anemia carrier. And a carrier may not show the symptoms of sickle cell anemia. He may lead a normal life. He may not show much symptoms of sickle cell anemia. So he can lead a normal life. Okay. Then another condition is, another genotype is HbS. That means the mutated allele comes in homozygous condition. Both the alleles becomes mutated. Or the person inherit the mutated alleles, both the alleles for that particular trait or for abnormal hemoglobin is mutated, and that person would be affected with sickle cell anemia, and he would exhibit all the symptoms of anemia, all the symptoms of sickle cell anemia. Okay. Now, another thing here is, very interestingly, sickle cell anemia is an example for codominance also. It's an example for codominance. We have already seen codominance, and the example we have seen then was AB blood group. That's an example for codominance we have studied. But sickle cell anemia or sickle cell anemia is also the carrier. Sickle cell anemia carrier is the best example, is another example for codominance. Here, the normal gene HbA is a dominant gene. The normal gene on the 11th chromosome is a dominant gene, and the mutated disease-causing allele HbS is also a dominant allele. So in a carrier with the genotype HbS, both the normal gene, both the normal dominant allele and the mutated dominant allele exist side by side, and they express their phenotype side by side. That means in such a person, with the genotype HbA, HbS, 50% of the RBCs would be normal and 50% of the RBCs would be sickle shaped. As 50% of the RBCs are normal, he may not exhibit much of the symptoms of sickle cell anemia. He may be able to lead a normal life. So remember that this is an example for codominance also. Now let us see the marriage between two carriers, sickle cell anemia carriers, and the genotype and phenotypes of the children. That means HbA, HbS. So let the man be HbA, HbS is a carrier. And the woman also is the carrier HbA, HbS. So as shown here in the diagram, their children are of three categories, three genotypes. Certain children can be of normal genotype HbA, HbA. They are normal. Their children also can be HbA, HbS carriers, and the children can be HbS, HbS, that is sickle cell anemia patient. Okay, so this is the pattern of inheritance of sickle cell anemia. Okay. Now let us go to another Mendelian disorder, Mendelian genetic disorder, that is phenylketonuria. Phenylketonuria. Phenylketonuria is also known as an inborn error of metabolism. Inborn error of metabolism. Phenylketonuria is due to an error in the metabolism. An error in the metabolism. So, what is the metabolism here? Here the metabolism is conversion of phenylalanine to tyrosine. What is phenylalanine? It is an amino acid. Phenylalanine is an amino acid. What is tyrosine? It is also an amino acid. It is another amino acid. That means one amino acid, phenylalanine, is converted, is transformed into, by metabolic pathway, in a metabolic pathway, it is converted into tyrosine, another amino acid. So this is a metabolic reaction. Conversion of phenylalanine into tyrosine is a metabolic reaction. And we had already studied that every metabolic reaction is catalyzed by enzyme. Every metabolic reaction is catalyzed by enzyme. So here also, the conversion of phenylalanine to tyrosine is catalyzed by an enzyme. And enzymes are proteins. And these proteins are synthesized by, or the synthesis of protein is managed, or these enzymes are managed by or directed by a gene. And here the enzyme is, that means the enzyme for the conversion of phenylalanine to tyrosine is called phenylalanine hydroxylase. The name of the enzyme is phenylalanine hydroxylase. And this enzyme phenylalanine hydroxylase converts the phenylalanine into tyrosine. That is very essential in our body. That metabolic reaction is very essential in our body. And the enzyme phenylalanine hydroxylase is produced by a gene. a gene that is situated on the chromosome number 12. Okay. So a mutation to this gene, a mutation happening to this gene situated on chromosome 12 makes the gene or converts the gene unable to produce the phenylalanine hydroxylase enzyme, or it may produce very less quantity of this enzyme. So either due to the absence of this enzyme or due to the presence of a less quantity of this enzyme, phenylalanine hydroxylase, conversion of phenylalanine to tyrosine would not take place. Or the reaction or the conversion may be very slow, as the case may be. As a result, what happens? This phenylalanine accumulates in the body, in the cells, or these metabolic activities are taking place in the cells. So phenylalanine, which failed to be converted to tyrosine, gets accumulated in the cells. And slowly, this phenylalanine would be converted to phenyl pyruvic acid. Phenyl pyruvic acid. That is not required in our body. But when the phenylalanine gets accumulated, that will be converted to phenyl pyruvic acid. Maybe for better elimination from the body. But this phenyl pyruvic acid gets accumulated. Phenyl pyruvic acid gets accumulated, especially when it accumulates in the brain cells. Brain cells will be damaged. And in children, if it happens in the children, of course it would happen in the children because it is an inborn error of metabolism. That would be there in the child because it is a genetic disease getting received from the parents. So in children, when the brain cells are damaged due to the accumulation of phenyl pyruvic acid, would lead to mental retardation. Not only mental retardation, associated with some other complications also would appear in the body. And this phenyl pyruvic acid, excess phenyl pyruvic acid, would be released through the urine also. So this condition is known as phenylketonuria. Phenylketonuria. Due to a mutation, it occurs due to a mutation of a gene located on chromosome number 12, which is responsible for the production of phenylalanine hydroxylase enzyme, which in turn is responsible for the conversion of phenylalanine to tyrosine. When phenylalanine is not transformed to tyrosine, it accumulates in the cells and slowly be converted to phenyl pyruvic acid, and the phenyl pyruvic acid accumulates in the cells, especially when it accumulates in the brain cells. That would lead to mental retardation in children. And also this phenyl pyruvic acid will be released through the urine also. And it may lead to some associated complications in the body also. So this is known as phenylketonuria. Okay. So these are some of the Mendelian disorders that we have to study. We shall wind up the class here. We will see in the next class.